بس لحظة شوي. ما أعرف ليه عم يعمل لي ترانسكريبشن كمان.
هذا لحاله بيفتح مع الريكوردينج.
طيب احنا قلنا انه this is the animal reproduction course. The first chapter is about the meiosis and the gametogenesis, and since we are talking about reproduction, we will talk about the gametogenesis, which is the production of gametes. And we always have during the production of gametes the phenomenon of meiosis. Okay, so that's why we should start by talking about the meiosis, and then we will talk about the other things later. So we were saying that we have during the gametogenesis we will generate gametes, which are the reproductive cells. And we have female and male gametes. The female gametes are called ova or eggs, and the male gametes are called sperm cells or spermatozoa. And now during the gametogenesis, we can have either the spermatogenesis, which is the production of sperms, and it takes place in the testes. And we have the oogenesis, which is the production of eggs, and it takes place in the ovaries. Now, during when we have the meiosis, we need the meiosis to generate the gametes. The meiosis only occurs in the reproductive cells. However, the mitosis occur in the somatic cells. Okay. Now we can see some. We can have also mitosis at some points, but the meiosis is restricted for the gametes or for the reproductive cells. Now, before that the meiosis starts, we should always start by a diploid cell. From a diploid cell, before the beginning of the meiosis, we should have a diploid cell which has 2 sets of chromosomes, 46 chromosomes, and each chromosome should have 2 chromatids. So before the beginning of the meiosis, as you know, in the cell cycle, we have the interphase that contains the G1, S, and G2 phases, and then we will have the meiosis or the mitosis in general. So during the S phase of the interphase, as you know, we will have a duplication of the DNA. And in this way, the cell will generate a second copy of its DNA content. So during the interphase, we will have, always during the interphase, we will have a duplication of or a replication of the DNA. The cell will become diploid, and then the meiosis can start. So always before the meiosis, before the start of the meiosis, the DNA should be replicated during the interphase. Okay. Once the DNA is replicated, we will have a cell having 2 N chromosomes, and each one of them has 2 chromatids. So we will have a cell having 2 sets of chromosomes, and each one of those chromosomes contain 2 chromatids, 2 sister chromatids. Now, the meiosis has 2 sequential divisions, the meiosis 1, which is directly followed by the meiosis 2. The meiosis 1 will allow the separation of the sister of the homologous chromosomes. So at the end of the meiosis 1, we will have 2 daughter cells, and in each one containing 1 one of the homologous chromosomes. Yani the homologous chromosomes, which were together in the diploid cell, will get separated in different cells, and in this way we will have a reduction of the number of chromosomes at the end of the meiosis 1, and we will obtain a haploid cell, so a cell or cells containing one set of chromosomes. And in the meiosis 2, the chromatids of each chromosomes will get separated. So during the meiosis 2, we'll have the separation of the sister chromatids. In this way, at the end of the meiosis, we will obtain 4 daughter cells, each one of them having one set of chromosomes. So we will have haploid cells, and each one of those chromosomes will have only one chromatid. Okay, so we'll start by a diploid cell, and we will end by four daughter cells. Each they are haploid and containing one chromatid. Now here, the meiosis, as we said before, this was the cell before the meiosis. This cell should duplicate its DNA content. So during the interphase, we will have a duplication of the DNA content. And now, once the DNA content, and before we had a diploid cell having only one chromatid for each chromosome, during the interphase, each chromosome will have 2 chromatids again. And now, once we have a cell having 2 chromatids for each chromosome and where the DNA is duplicated, we can start the meiosis. The meiosis 1 is called a reductional division because as a consequence of this meiosis, the number of this first step of the meiosis, the number of chromosomes will be reduced to the half. And if we had 46 chromosomes at the beginning of the meiosis, by the end of the meiosis 1, we will have 23 chromosomes, each one having 2 chromatids. Okay. So this is why the meiosis 1 is called a reductional division. And during the meiosis 1, as we are going to see, we will have the crossing over phenomenon also, but we will talk about this just now. And now in the meiosis 2, once the meiosis 1 is completed, directly start the meiosis 2. We don't have a duplication of the DNA between the meiosis 1 and the meiosis 2. The duplication of the DNA occurs only before the meiosis 1. So before the entry of the cell in the meiosis. Okay, but after the meiosis 1, the cell directly enters in the meiosis 2. And during the meiosis 2 is like a mitosis. We have a separation of the sister chromatids. So here we have a separation of the sister chromatids. And as a result of the meiosis 2 is called an equational division. As for the mitosis is also an equational division, which means equational division means we have an equal number of chromosomes as the parent cell. So in the meiosis 2, we start by a haploid cell. We obtain each cell give 2 daughter cells which are also haploid, but they have one chromatid only. So the meiosis 2 is called an equational division, and the meiosis 1 is a reductional division. Now, we have said also that in the meiosis 1 and 2, each we have the prophase, metaphase, anaphase, and telophase. And the prophase of the meiosis 1 is divided into 5 parts also. So the prophase 1, the prophase of the meiosis 1 is the longest phase of the meiosis, and it is subdivided into 5 steps. And those are the different steps, and they are different, or we can They are subdivided according to the state of the chromatin during those different stages. The first stage is called the leptonema. Then we have the zygonema substage of the prophase 1. Then we have the pachinema. Then we have the diplonema and the diakinesis. So what happens during those different substages of the prophase 1? As you know, during the prophase, we have a compaction of the DNA or of the chromatin. It will get more dense or more compact. Okay, and at the end of the prophase, we will have a disappearance of the nuclear envelope. But here we are going to see during the or in the different steps of the prophase 1, what's going to happen, so we can reach the metaphase. So during the leptonema is the first substage of the prophase 1. It is called so leptonema. It's come from the Greek leptos, which means thin. So here the chromatin will start to condense and will appear as thin threads in the nucleus. So whenever we put a dye that interacts with the chromatin, we can observe the chromatin as thin threads inside of the nucleus. We can see here those thin threads, okay? So the nuclear chromatin begins to condense during this substage, and also the telomeres, the telomeres are the tips or the ends of the chromosomes. So the telomeres will bind to the nuclear membrane. They will bind to the nuclear membrane. In this way, the chromosomes will get attached to this nuclear membrane through the attachment plate. So we have a complex of proteins which is called the attachment plate. This complex of proteins will enable the binding of those or of this chromatin or of those chromosomes to the nuclear envelope. Okay, so during the substage also, the telomeres will bind to the nuclear envelope through the attachment plate. And then those chromosomes attached to the nuclear envelope will move to one end or to one pole of the nuclear envelope. So they will assemble at one end or at one side of the nucleus, and in this way they form a sort of bouquet. Okay, so we call them a chromosome bouquet. So at the end of the leptonema, and directly before the entry of the cell in the second substage, which is the zygonema. So at the leptonema-zygonema transition, we will observe the formation of the chromosome bouquet, where all the chromosomes will gather at one side of the nucleus, or on one side of the nuclear envelope. We are going to see why it is important for them to gather at one side of the nuclear envelope. Okay, like here you can see we don't have the chromosomes dispersed all over the nuclear envelope. This is the nuclear membrane, okay, but we can see them, all of them here. Okay, and here also, this is the nuclear envelope, and we can see them attached here. And they look like thin threads also. So this is the leptonema, and the appearance of the chromosome bouquet happens at the leptonema-zygonema transition. Now, the second substage of the prophase 1 is the zygonema. And zygon means in Greek paired, okay? So here, during this substage, the homologous chromosomes will get paired or will get attached to each other. Okay, because as you know, as we have said in the meiosis and the meiosis 1, during the first meiotic division, we should have a separation of the homologous chromosomes. So how it is possible for them to get each one of them separated in a different cell? To get separated, they should attach to each other first, and then we will see why this attachment is important, and then this will allow them to correctly separate into two different cells. So they should find each other, and then this will make it possible for them to separate in two different cells. So when they are arranged or assembled near to each other here at one side of the nucleus, this will allow them to get to be near. This will allow the chromosome to get close to each other, and then for each chromosome to find its homologous pair. Okay, it will make it easier for them to find their homologous pair. So at the zygonema, the chromosomes or the homologous chromosomes will begin to pair, and this is called the pairing event is called the synapsis, which means the union. So they will get united or assembled to each other. Like here you have one pink and one maternal and one paternal chromosome. They are homologous chromosomes, so they will get assembled to each other. And this assembly or this synapsis, so this synapsis between the homologous chromosomes is made possible due to a complex of proteins called the synaptonemal complex. So this is synaptonemal from synapse. So this is a complex that will allow the attachment or the binding of the two homologous chromosomes. Okay, like those white lines here, they are the complex of proteins, the synaptonemal complex, binding the maternal to the paternal chromosomes. Okay, so the synapse chromosomes are held together by a complex called the synaptonemal complex, and this complex is formed between two sets of sister chromatids in each bivalent. In fact, once they are held or synapsed together, we call them a bivalent. Bivalent means two. So we have two chromosomes held together. We call them a bivalent, or we call them also a tetrad. A tetrad means 4 chromatids. So we can say a bivalent for 2 chromosomes held together, or a tetrad because we mean 4 chromatids held together, okay, or synapsed together. So we call this those homologous chromosomes synapsed, a bivalent or a tetrad. Now this synaptonemal complex, we can see them here, one chromosome in blue and its partner in red. And we have the synaptonemal complex, the white bars between them, holding them together, and this is the synaptonemal complex. This synaptonemal complex is composed from 2 transverse or 2 lateral elements, okay, and 1 central element. That's why we call the synaptonemal complex a tripartite, 3-4-3. So it is composed from 3 parts, 2 lateral elements and 1 central element. And here you can see that we have this red chromosome, which is, let's say, the maternal chromosome, and this is the paternal chromosome, and they are held together by those lateral lines, which are the synaptonemal complex, okay? And we have here this middle line. It's the central element of this complex, of the synaptonemal complex. And this is here, this is picture also of synapse chromosomes. Here we have 2 homologous chromosomes, one here and one here, and we have the synaptonemal complex in the middle. We have the lateral elements and the central element, okay? And the synapses begins near the telomeres, begins near the telomeres, and it grows like a zipper toward the center. So it grows from the extremities and moves toward the center. Okay. So this is the synapses. The synapses occurs during the zygonyma, yani during the second substage of the prophase 1. And at the end of the zygonyma, the synapses is complete for all of the chromosomes. So all of the homologous chromosomes will get synapsed, and the synapses is complete. And then the cells can enter the third substage of the prophase 1, which is the Pachytene. Okay, once the synapses is complete, we say that the cells enter the Pachytene substage. So by the beginning of the pachynyma or the Pachytene stage, the synapses is complete. Okay, and the word pachynyma comes from pachos in Greek, which means thick. So here during the pachynyma, we will observe thick threads of chromatin and not thin threads of chromatin. Okay, during the leptonyma, if you remember, we had thin threads because the chromatin started to condense here. Now here, since they are paired, the homologous chromosomes are paired and condensed, they will appear as thick threads and not anymore as thin threads. So during this pachynyma stage, we can call it also Pachytene. Okay, thick threads of chromatin form due to the synapses. So due to the assembly of homologous chromosomes, due to the synapses, we will observe thick threads of chromatin. During this stage, we will have crossing overs along the chromosomes. Do you know what are the crossing over?
Yes.
sharing the parts of chromosomes between
Yes,
homologous chromosomes.
yes.
Non-sister chromosomes of homologous pair.
Non-sister chromatids of homologous pairs. Okay. During the pachynyma stage, we will have the crossing over between non-sister chromatids of homologous pairs. Okay. So we will have the exchange of genetic material or of alleles between the homologous chromosomes. And this will allow, why it is important, why is the crossing over important during the meiosis?
Diversity.
Yes, it will create a diversity. This will create different in the gametes, different chromosomes, different from the paternal ones. Okay, because they have new fragments coming from another one. Okay, so this will generate recombinant chromosomes. So chromosomes having new segments or new alleles that were not present in the paternal cells. Okay, and which will make those gametes not, those daughter cells coming from the parent cell not genetically identical to the parent cell. Okay, and this will allow for the diversity of the offsprings or of the individuals later. Okay, since they will have a genetical material different from their parents. And we'll see this also later. Now, during this stage, we will have crossing over. To have crossing over, look here, between the two homologous chromosomes, we have like those X or what do we say, intersection points between the two homologous chromosomes, the paternal and the maternal one, and between the two non-sister chromatids, we'll have cross points or an intersection points. And those intersection points, we will have the crossing over between the fragments between those intersection points here. which will allow also the crossing over. Now, this crossing over is made possible once due to the synapses, because if we don't have the synapses, they couldn't be they couldn't get close to each other, and we couldn't get this crossing over, and also due to the presence of recombination nodules. So before the occurrence of the crossing over, we have a complex of proteins that should bind to the synaptonemal complex. Okay, this complex of protein is called the recombination nodules, and depending, we will have the crossing over directly next to the adjacent to the sites where those recombination nodules bind. Those recombination nodules, they are proteins. Next to them, we will have the crossover. So they will allow the crossing over to happen. Okay, so they are essential for the occurrence of the crossing over. So during the parenchyma, we will have thick threads of chromatin that form due to the synapses, and we will have recombination nodules that will come and will associate at different points of the synaptonemal complex. Those recombination nodules will associate at different points of the synaptonemal complex, and at the places where they associate, directly adjacent to them, we will have the crossing over. Okay, we can have a crossing over. And the crossing over occurs at different points of the synaptonemal complex. The crossing over will allow the exchange between non-sister. Yes.
شو أهمية الكروسنج أوفر؟
مثل ما قلنا إنه بخلي يصير في عند الكروسنج أوفر، يعني we will have يعني one part of the chromatids will be cut, okay, and they will be exchanged. We will have an exchange of alleles or fragments of DNA between the non-sister chromatids of the homologous chromosomes. And the paternal chromosome, let's say here, will get a red fragment from the maternal chromosome, and also the maternal chromosome will get a fragment from the paternal chromosome. So this will create chromosomes that are different from the paternal ones, from the initial ones, okay?
Okay.
And this will create diversity in the gametes and as a consequence in the offsprings. رح نشوفهم، نرجع نشوفهم بعدين.
دكتور هلا الكروسنج اوفر في يصير مع التو سيستر كروماتيدز بذات وقت؟
Here, no, it will happen only between the non-sister chromatids. Okay.
لا أنا I mean صح إنه بالهومولوجس بير، بس أنا عندي بالكل هومولوجس بير، يعني بكل بير عندي تو سيستر كروماتيدز، معقولة اثنيناتهم يعملوا كروسنج اوفر مع الهومولوجس كروموزوم؟
قصدك the two chromatids, the two sister chromatids بيعملوا cross over مع التاني؟
Yes.
لا، وحدة only one strand will make the crossing over with the homologous chromosome. Okay.
يعني هيدا الكومبلكس الساينابتيك كومبلكس هو بس بيصير على بين one sister chromatid each pair.
Yes, yes.
Okay, deal. Thank you.
So we will have an exchange between as a result of this crossing over, we will have an exchange between non-sister homologous chromatids. Okay, this is the allele exchange. So whenever we have an exchange of DNA fragments between the non-sister homologous chromatids, we will have an exchange of alleles. Okay, and this is called the intrachromosomal shuffling or mixing. Okay, so the crossing over is called an intrachromosomal mixing, or will allow an intrachromosomal shuffling. Okay. Intrachromosomal means within. Intra means within. So intrachromosomal shuffling means within the same pair of chromosomes. Since here we have a pair of homologous chromosomes, so this mixing will be happening inside of this pair. This is called the intrachromosomal shuffling. Now, the points where the crossing over occurs, you can see here the physical points where the two chromatids, where the two non-sister chromatids will intersect. This point will be called the chiasma, okay? And the plural is chiasmata. So at those intersection points between the two different chromatids, the two non-sister chromatids, are called the chiasma, okay, or the chiasmata. So during a crossing over, as we said, we have an attachment of, or the association of the recombination nodules to the synaptonymal complex, and we have this intersection between the two non-sister chromatids that is essential for the crossing over. So this intersection is called the chiasma. Those points, the points of intersections are called chiasmata. Those chiasmata are important for the happening of the crossing over. So we need those intersection points for the crossing over to happen, okay? So during the pachynyma, we can observe intersection points between the non-sister chromatids. Those intersection points are called the chiasmata or the chiasma. So at the chiasma occurs the fusion or the intersection and exchange of genetic material. Okay. So this is regarding the pachynyma, and then we will have the diplonyma. The diplonyma, diplo from di, which means two. So here we have two threads. Okay, so at this substage of the prophase 1, the homologous chromosomes, before we have said that they are completely synapsed. It's like they are completely attached to each other and appearing here as thick threads. Here we will start to see two threads. We're not seeing anymore one blue and let's say blue and red threads attached to each other. No, we can start to see two threads. Why? Because the synapses between those homologous chromosomes will start to disappear or to degrade, okay? So at this stage, the synaptonymal complex will start to disappear. It will not be maintained all along of the chromosomes. It will not hold all the chromosomes together or the homologous chromosomes together anymore. It will start to disappear, but it will be conserved at certain point, okay? So that's why at this stage, when we observe the chromosomes under the microscopes or the cell or the nucleus under the microscope, we can observe two threads, okay? So here the synaptonymal complex disappears and homologous chromosomes begin to separate since there is no more synaptonymal complex. But they remain, those homologous chromosomes will not separate completely. They will remain tightly bound at chiasma, at the chiasma points. Okay, like we said here, we have the formation of chiasma points. Those chiasma points will be maintained. Okay, we always have a synaptonymal complex at the level of the chiasma, but in the other places of the chromosome, the synaptonymal complex will disappear. So here we have in this phase the disappearance of the synaptonymal complex, and we'll still have those two homologous chromosomes bound together at the level of the chiasma, okay? And during this stage, we will have more condensation of the chromatin or of the chromosomes. They will get more condensed also, and
راح الصوت.
إيه.
طلعت من الميتنج دكتور.
بلكي راح الإنترنت عنده.
انقطع الإنترنت من عندي. meeting. Okay. So each chromosome now at this diploidy state. Yes.
الشاشة بايظة ما يعني ما مش طالعة.
Okay. هلا
أنا
بعد ما طلعت.
إذا الإنترنت بطيء بدها شوية وقت لتبين.
ما بعرف هلا إذا بيني تقولوا لي. بعد ما بينت؟
لا
بعد ما بين.